NCERT based single sentence questions, cell cycle and cell division. Let's start. Characteristics of cells which are indeed of all living organisms are growth and reproduction. All cells reproduce by dividing into two with each dash cell giving rise to two dash cells each time they divide. Each parental cell giving rise to two daughter cells each time they divide. Very important process in all living organisms is cell division. The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell and eventually divide into two daughter cells is termed as cell cycle. Although the cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. Replicated chromosomes are then distributed to daughter nuclei by a complex series of events during cell division. And these events are themselves under genetic control. A typical eukaryotic cell is illustrated by human cells in culture. Cells divide once in approximately every 24 hours. Duration of cell cycle in every organism vary. Time taken for cell cycle in yeast 90 minutes. Two basic phases of cell cycle are interphase, two basic phases, interphase and M phase. Phase which corresponds to the interval between mitosis and initiation of DNA replication, G1 phase. If the initial amount of DNA is denoted as 2C, then it increases to 4C. Even after S phase, there is no increase in the chromosome number. Yes, true. In S phase, amount of DNA per cell doubles, but there is no increase in number of chromosomes. True. In S phase, amount of DNA doubles, which means diploid chromosomal set become tetraploid. False. In animal cells, during the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. G2 phase. In animal cells, during the dash phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. S phase. If the cell had diploid or 2n number of chromosomes at G1, even after S phase, the number of chromosomes remains the same, that is 2n. True or false? True. Some cells in adult animals do not appear to exhibit division, which remains in G0 phase. What are the cells? Heart cells. Cells that do not divide further exit G1 phase to enter an inactive stage called G0 of the cell cycle are called as quiescent stage. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so depending on the requirement of the organism. That is quiescent stage. In animals, mitotic cell division is only seen in the diploid somatic cells. Animals who undergo mitosis for the formation of gametes, honeybee. Plants can show mitotic divisions in both haploid and diploid cells. True. Animals can show mitotic divisions in both haploid and diploid cells. False. Most dramatic period of the cell cycle involving a major reorganization of virtually all components of the cell is M phase. Since the number of chromosomes in the parent and progeny cells is the same, Mitosis is also called as equational division. Cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. True. Division of nucleus is called karyokinesis. Division of cytoplasm is called cytokinesis. The four stages of nuclear division are prophase, metaphase, anaphase, telophase. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, that is karyokinesis, but the cell itself is divided into two daughter cells by the separation of cytoplasm called cytokinesis, at the end of which cell division gets completed. In animal cell, cytokinesis is achieved by the appearance of a furrow, in the plasma membrane. There is a furrow appears in the plasma membrane. The dash gradually deepens and ultimately joins the center, dividing the cell cytoplasm into two, that is called cytokinesis, that is in animal cell, that is furrow. Plant cells, however, are enclosed by a relatively inextensible cell wall. In cell wall, therefore, they undergo cytokinesis by different mechanism. In plant cells, the formation during cytokinesis starts in the center, of the cell and grows outward to meet the existing lateral walls. The formation of the new cell wall begins with the formation of a simple precursor called the cell plate that represents the middle lamella between the walls of two adjacent cells that is in plant cells. Status of mitochondria and plastids during cytoplasmic division get distributed between the two daughter cells. Liquid endosperm in coconut is an example for syncytial condition. If karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises, leading to the formation of syncytium. Phase between two successive M phases. Interphase. Average duration of cell cycle in human cell, 24 hours. Average time duration of cell division in cycle of human cell, 1 hour. The phase when the actual cell division or mitosis occur, apparent phase or division phase. Phase which represents the actual cell division or the division or mitosis occurs, that is M phase. Phase lasts more than 95% duration of cell cycle. Interphase. Phase which is completed in 1 hour. M phase, mitosis. Phase starts with nuclear division, mitosis. Separation of daughter chromosomes called karyokinesis. Separation of daughter chromosomes is called karyokinesis. Division of cytoplasm is called cytokinesis. Phase called as resting phase. Interphase. Phase corresponds to the interval between mitosis and initiation of DNA replication, that is G1 phase. Phase considered as preparing for cell division, that is interphase. Phase marks the period which DNA synthesis or replication takes place. S phase. First phase of karyokinesis, prophase. Phase both cell growth and DNA replication occur. Interphase. New DNA molecules formed are not distinct but intertwined in S and G2 phase. Initiation of condensation of the chromosomal material, prophase. Phase DNA replication begins in the nucleus, S phase. Initiation of condensation of chromosomal material, Prophase. Phase cell is metabolically active and continuously grows but does not replicate its DNA. G1 phase. Centrosome undergo duplication. S phase. Phase during this time, the amount of DNA per cell doubles. Centrosome begins to make towards opposite poles of the cell. Prophase. Phase centriole duplicates in the cytoplasm. Phase centriole duplicates in the cytoplasm. S phase. Phase centriole duplicates in the cytoplasm. That is S phase. Proteins are synthesized in preparation for mitosis while cell growth continues. G2 phase. G2 phase. Chromosomal material condenses to form compact chromosomes. Prophase. Complete disintegration of the nuclear envelope. Metaphase. Chromosomes are seen to be composed of two chromatids attached together at the centromere. Prophase. Each centrosome radiates out microtubules called as asters. Prophase. Two asters plus spindle fibers, mitotic operators. Prophase. End of this phase, no Golgi complex, endoplasmic reticulum, nucleus, nuclear envelope. Prophase. Chromosomes are spread in the cytoplasm of a cell. Metaphase. Nucleolus, Golgi complex, ER. Reform. Telophase. By this stage, condensation of the chromosomes completed. Metaphase. At the time of division, organelles like mitochondria, plastids get distributed between in the two daughter cells between, that is in cytoplasmic division. Cytoplasmic division. If karyokinesis occurs, but cytokinesis does not occur, it results. Karyokinesis occurs, cytokinesis does not occur, it results multinucleate condition. Multinucleate condition. Chromosomes can be observed clearly under microscope, that is in metaphase. Reason for syncytial condition. Karyokinesis occurs but not cytokinesis. Morphology of chromosomes easily studied at metaphase. Liquid endosperm in coconut is an example for syncytium. Chromosomes made up of two sister chromatids. Metaphase. Nuclear envelope develops around the chromosome clusters at each pole forming two daughter nuclei. Telophase. Centromeres split and chromatids separate. Anaphase. Chromatids move to opposite pole. Anaphase. Two sister chromatids held together by centromeres. Metaphase. Division of nucleus. Karyokinesis. Division of cytoplasm. Cytokinesis. Chromosomes cluster at opposite spindle poles and their identity is lost as discrete element. Telophase. All the chromosomes coming to lie at the equator. Metaphase. Stage nucleolus, Golgi complex and ER reform. Telophase. Formation of new cell wall begins with the formation of simple precursor called cell plate. Kinetochore of chromatid connected to spindle fiber. Metaphase. Cell plate that represent between the two plant adjacent cells. In the plant, cell plate that represent between the two plant adjacent cells. Present between the two plants of adjacent cells, that is cell plate. Spindle fibers attached to the kinetochores of chromosome, that is metaphase. Next, we'll continue with meiosis, reductional division. Reductional division is called meiosis. Reductional division is said to be stages of prophase 1, leptotin, zygotin, pakitin, diplotin, diakinesis. Reproductive cell division. Reproductive cell division is nothing but meiosis. Interkinesis. Interphase without S phase. Interkinesis means interphase without S phase. We call interkinesis. Heterotypic division. Heterotypic division is meiosis 1. Number of haploid cells formed at the end of meiosis 2, 4. Examples of homotypic are equational division, mitosis and meiosis 2. Largest phase of meiosis 1, prophase 1. Synaptonemal complex is formed at zygotin. Synapsis, process of pairing of homologous chromosomes is called synapsis. Process of homologous pairing of chromosomes is called synapsis. Synapsis means process of pairing of homologous chromosomes. Process of pairing of homologous chromosomes is called synapsis. Bivalent or tetrad. Bivalent or tetrad means pair of synapsid homologous chromosomes are called bivalent or tetrads. Recombinase. Recombinase enzyme involved in recombinase is the enzyme involved in crossing over. Appearance of recombination nodules in paketing stage. Crossing over occurs. What is crossing over? Crossing over means exchange of genetic material. Crossing over means the exchange of genetic material between two non-sister chromatids. Between two non-sister chromatids of homologous chromosomes. Between non-sister chromatids of homologous chromosome. Synaptonemal complex is composed 3 thick lines of DNA. Synaptonemal complex is composed by 3 thick lines of DNA and proteins. Dissolution of the synaptonemal complex or desynapsis done at resolution of synaptonemal complex or desynapsis done at diplotin. X-shaped structure formed during crossing over is called chiasmata. Chiasmata visible in diplotin stage. Diplotin can last for months of years in oocytes of some vertebrates. The final stage of prophase 1 is diakinesis. Final stage of prophase 1 is diakinesis. Terminalization of chiasmata at diakinesis. In animals, spindle fibers are formed by centrioles. In plants, spindle fibers are formed by microtubules. Aster, that is centriole plus chromosomes plus spindle fibers. They say asters. Myocyte, cells which undergo meiosis are called myocyte. In algae, myocyte is zygote. In bryophyte, pteridophyte flowering plants, myocyte is spore mother cell. 2n changed into n in meiosis. N changed into 2n in syngamy. Haploid changed into diploid in syngamy. Anaphase 1 is characterized by segregation or disjunction of chromosomes. Segregation or disjunction of chromosomes. Meiosis is responsible for forming dash phase in the life cycle, haploid phase. Bringing variations in progeny is due to meiosis. Dyad condition is seen in telophase. Dyad condition is seen in telophase. The stage between meiosis 1 and meiosis 2 is called as interkinesis. Meiosis 2 is equal to mitosis. Division of centromere is absent in anaphase 1. Meiosis 2, division of chromosomes at centromere. Division of chromosomes at centromere is absent in anaphase 1. Meiosis 2 differ from normal mitosis. Meiosis differ from normal mitosis. The two chromatids of a chromosome are dissimilar, and daughter cells produced after meiosis 2 are not similar to each other, are mother cell. The stage is marked by terminalization of chiasmata, diakinesis. Number of spindle apparatus formed in meiosis 3. That one in metaphase 1 and of meiosis, and metaphase 2 in metaphase 2. All spindle fibers attached to chromosome is called spindle apparatus. Number of spindle apparatus formed in mitosis 1 at metaphase of mitosis. Stage the chromosomes become gradually visible under the light microscope. Leptotin. Compaction of chromosome continues throughout the stage. Leptotin. During this stage, chromosomes start pairing together, and this process of association is called synapsis. Such paired chromosomes are called homologous chromosomes, that is in zygotin. Electron micrographs of this stage indicates that chromosome synapsis is accompanied by the formation of complex structure called synaptonemal complex, zygotin. The complex formed by a pair of synapsed homologous chromosomes is called bivalent or tetrad. Zygotin. Bivalent or tetrad, these are not more clearly visible at this stage. Bivalent or tetrads, these are more clearly visible at this stage. More clearly visible at packeting. The two stages of prophase 1 are relatively short-lived compared to the stage packeting, leptotin and zygotin. During this stage, the four chromatids of each bivalent chromosome become distinct and clearly appears as tetrads, that is packeting. This stage is characterized by the appearance of recombination nodule, the sites at which crossing over occur between non-sister chromatids of the homologous chromosome, packeting. Crossing over is the exchange of genetic material between the two homologous chromosomes, packeting. Crossing over leads to the recombination of genetic material of the two chromosomes, packeting. Recombination between homologous chromosomes is completed by end of this stage, leaving the chromosome linked at the sites of crossing over, packeting. In the beginning of this stage, recognized by the dissolution of the synaptonemal complex and the tendency of the recombined homologous chromosomes of the bivalent to separate from each other except at the sites of crossover. These X-shaped structures are called chiasmata, that is diplotin. Final stage of meiosis, prophase 1 is diakinesis. The stage which is marked by terminalization of chiasmata, diakinesis.